Okay, all right. So now, once you make PEP, right, from gluconeogenesis, the first step we did, once you make PEP, then what will happen? All the reversible steps will continue using the same enzyme and the product of the same coenzyme in the reverse direction, right? Until you hit the other irreversible step, which was what? So F16BP, right? Fructose 1,6 bisphosphate. So how do we handle that? We need to use some enzyme. So F16BP, right? So that's our F16BP. And now we need to do something to go back to what? F6P, right? So what do we do? What enzyme did you use to make this? So we use a kinase, right, to make it. Now we need something else to break it, right? And these enzymes, so kinase will phosphorylate, right? But phosphatase, they hydrolyze the phosphate ester bond, right? So they dephosphorylate by hydrolyzing that phosphate ester bond, right? So what product we're going to get from here? So we get back F6P, right? That's what we're going to get from this. And there are different isoforms of this enzyme. So this will be called fructose bisphosphatase 1, FBPS1. So kinases and phosphatases do opposite reactions, right? One will phosphorylate, the other one will dephosphorylate. So that's it. We have our second bypass, right? We have F6P. So now what will happen? What has to happen from here? But we got stuck again at G6P, and we need to make glucose. So G6P. So this will be the second bypass, and so the first was, of course, going from pyruvate to PEP, right? We use two different enzymes. But now second bypass and the third bypass is we have to make glucose from G6P. Right? And we need to make glucose. So just get OH back. Okay, so that's your glucose and G6P. So what enzyme we're going to use in this? Phosphatase, right? Phosphatases will hydrolyze this bond PI, phosphate ester. So we will just use glucose 6 phosphatase, GLC 6. Okay, so now once we have glucose, then you can feed this to brain, right? Or you can give this to our red blood cell, RBC, because they survive on glucose as fuel, right? The next thing I was going to discuss is that Gluconeogenesis is expensive. Yeah, we do need ATP, right? And well, remember we use GTP, and it's kind of like, so we need, which is also a high energy compound, right? So we need high energy compound GTP. You can think of that kind of similar to ATP. So we really need energy to make this happen. So it is expensive but necessary. And so gluconeogenesis happens. So gluco, this happens when our glycogen storage is depleted, right? So this happens. And where is where is glycogen stored? Right? So glycogen is stored in the liver, also in the muscles. We have glycogen, but once mainly the liver glycogen is used up when you're starving, right? If you eat an early dinner, for example, probably if you have, by next morning, all the glycogen storage is almost gone, right? So then, but if you fast for prolonged amount of time, then body will find ways to generate glucose from other resources, right? And so that the brain can use it. Metabolic conditions we discussed will be starvation, right? Starvation, exercise. If you do vigorous exercise, right, then we do gluconeogenesis. All right, now you can also get glucose from amino acids, right? So, and we'll learn them later in the semester. And so there are some amino acids called glucogenic amino acids. And so you get glucose from glucogenic amino acids, right? Not all, but some amino acids will give you. All right. Now the other thing is, what about fatty acids? Can you get glucose from fatty acids? Acetyl-CoA, right? So acetyl-CoA, um, remember, if you have acetyl-CoA, that is one-way path, right? Going from pyruvate to acetyl-CoA, that's one way. So from acetyl-CoA, we cannot come back. And a lot of the fat is going there. So how do we use fatty acids to get glucose? Yes, glycerol. So fatty acid part cannot make glucose, but the glycerol part, so for example, TAG, right? We have learned a lot about TAG. What is the full name of TAG? Triacylglycerol. So we get, remember the fatty acid part, the tail. This was the tail, right? Fatty acid. And we made, last semester we made ester bond, right, with glycerol to make TAG. And so this will be hydrolyzed to make free fatty acids and glycerol. So this glycerol can make glucose by gluconeogenesis. So fatty acid part tag from here, the fatty acids, these actually cannot make glucose, right? But the glycerol part can make glucose. So most of the gluconeogenesis is happening in the liver, but some also in the kidneys. So approximately about 60 to 70% in the liver, and the rest is in kidneys. So about 30 to 40%. Okay. All right. So gluconeo, part of it actually partitions between cytosol and mito matrix. Mito matrix is the abbreviation for mitochondrial matrix, which we'll use a lot in this semester. And this is in the cytosol, right? So the pyruvate actually has to enter mitochondria. And so here we have, if this is our mitochondria, it has, so you need to draw it bigger to show all the things that are happening inside it. Okay, so and it has this thing, right? All the cristae and whatnot inside. So that's your mitochondria. Pyruvate enters, and normally this will happen, right? You get acetyl CoA. Right? Normally, we haven't done that yet, but and this will be part of the citric acid cycle, CSE. And there are two intermediates here which I'll write. One of them is malate, and the other one is OA. What is malate? We haven't done it yet. I don't expect you to know. But we have done OA. What is OA? We looked at OA last class, right? Is it? So we added the carboxylic acid group, remember, to pyruvate. So that's our OA. So that's OA. So normally this is what will happen. Under normal metabolic conditions, the glucose will go to pyruvate, it will go to acetyl CoA, then citric acid cycle. We get a bunch of NADH, ATP from here. But when you are doing gluconeogenesis, then you are trying to make pyruvate go the other direction, right? Right? You're trying to make glucose from pyruvate. So this process has to slow down somehow, right? So the way to do that is to siphon some of the intermediates from here back into the gluconeogenesis. So why does it has to happen between mito and so here pyruvate carboxylase, right? That was one of our enzyme. Which we use in gluconeogenesis. Unfortunately, this enzyme is not present in the cytosol. It's only present in the mitomatrix. So this step cannot happen in the cytosol. So pyruvate carboxylase, we use it to make OA, right? And this is only present in the mitochondria. So pyruvate has no option other than entering mitochondria to make OA, right? So here, let me do here, pyruvate enter mito, then go to acetyl CoA. So that's your normal. When gluconeogenesis is happening, this pyruvate will actually go to OA. You make OA from pyruvate by this pyruvate carboxylase, right? Remember, this is a mito enzyme, so it has to happen in the mito. Then in gluconeo, what happens after OA? What happens to OA? We make PEP by PepsiK, right? So that is cytosolic enzyme. PepsiK is a cytosolic enzyme. So we need to transport OA from here back out into the cytosol so that we can make PEP, right? So here from OA we made PEP, remember? OA went to PEP and the enzyme was PepsiK or PEP carboxykinase. But this happens, this is the cytosol. So two different steps are happening in two different places. The first one is in the matrix, the other one is in the cyto. Question? Oh, sorry. Supposed to be pyruvate. So OA can come out, right? OA can simply come out, right? And you can make gluconeogenesis happen, right? But because I'm saying it, so there must be a twist. So it turns out there's no OA transporter. So OA cannot simply come out. So it has to be converted to something else. Then it can come out. And what could that be? I wrote malate for a reason, right? So OA will go back to malate. Now, once I draw the structure, you will see how that happens. So OA will go back to malate. Okay. And here, see, anyone remember the structure of OA? 4 carbons, okay. So how do you start? CO2 minus, okay. That's our way. And malate is this. So what happened from pyruvate to lactate? What did you do? Pyruvate to lactate. Reduce the carbonyl group. Same thing we'll do here. So now this is our malate. We just reduce the carbonyl group to OH. Now already based on what you have learned, you can predict what type of enzyme will do it. Which type? Dehydrogenase, right? So this is malate dehydrogenase. And which coenzyme do we need? NADH, because it's reduction, right? So we need NADH, then you make NAD plus. So NADH plus H plus, we make NAD plus. Because it's reduction, right? OA to malate is reduction, we reduce that carbonyl to alcohol. So we need NADH, not NAD plus. From here, so we do malate dehydrogenase, right? And we use NADH. Make NAD plus. And so now malate can escape. And I will make it escape on this side of the board. So now malate in the cytosol, it moved from mito to cyto. I'll use the orange chalk to show the gluconeogenesis. But is malate part of gluconeogenesis? Is it? So what must happen to it? Go back to A. Okay. So we are playing ping pong, right? So malate will go back to A. Again, malate dehydrogenase. This was done by mitochondrial malate dehydrogenase, right? So here I will take a note that this step is done by mitomalate dehydrogenase, right? But that enzyme pool is kept separate from cytosolic malate dehydrogenase. Um, so now what we need here from malate to go from malate to A? What enzyme? Same malate dehydrogenase, right? Because these are reversible enzymes. And what else? Do we need NAD plus or NADH? NAD plus. And go to NADH. Okay, so now we are back on track, right? Way, same way now is in the cytosol. Now we can go back. So go to PEP, right? And this will go on to make glucose. Just to recap, what happened here? We made pyruvate in glycolysis, right? Pyruvate, then under gluconeogenetic condition, we want to convert pyruvate back to glucose, right? And all of glycolysis happened in the cytosol. But the first step of gluconeogenesis is to make way from pyruvate by this pyruvate carboxylase, which is mitochondrial enzyme. It is not found in the cytosol, right? So that is why pyruvate has to go back to OA, and this must happen in the mito because the enzyme is only found in the mito. And OA could simply come out, but there is no OA transporter. So a clever way to do it will be to convert OA to malate in the mitochondria. Then malate can come out. And once malate is out, then you can make OA, right? And go to glucose. Now what happens? See, the malate is also an intermediate in the citric acid cycle, right? We haven't learned it yet, we'll learn. But see, I drew this circle and malate and OA. So malate is an intermediate in the citric acid cycle. So if you siphon malate out of the mitochondria and CSE is happening in the mitochondria, so what will happen to the CSE? Slow down, right? So it will slow down. So this is another mechanism by which we slow down citric acid cycle and use the resources to make glucose. There's another point, couple of more points. This NADH. Do you think there is a bigger reason why this has to happen in the cytosol? Not bigger reason. There is another benefit of happening this in the cytosol. So when we made glycolysis, did we use NAD plus? Yes. What did we make? NAD plus goes to NADH. So in one of the steps, we did use NAD plus to make NADH. That was a reversible step. If you want to go back, what do you need? NADH. So this NADH is used up to make that step happen. So this NADH is used up in gluconeo. Right? There was that step, another dehydrogenase. What dehydrogenase was it? Do you remember? GAP dehydrogenase, right? Yes. So this is where it will be used. The other reason is that the cytosolic NADH concentration is very low. Okay. So it's actually it's very low. So it's very low. So if you wanted to do gluconeogenesis normally in the cytosol, you won't have NADH to make it happen, right? But by doing this way, you also lift that limitation. So now you have NADH to make this happen. So the way to study these things, pathways actually is to draw out all the steps, right? Just the way I do it on the board, draw out all the steps, write all the enzyme names, write all the coenzymes, and use the logic for example, the redox reactions. Is it oxidation? Is it reduction? Should you be using NAD plus? Should you be using NADH? These are the logics that we need to think about. So the take home, I guess the big picture or the key points from gluconeogenesis is that first of all, the first enzyme is not available in the cyto, so it has to happen in the mitomatrix, right? And so pyruvate carboxylase will convert pyruvate to OA. It would have solved the problem if OA simply come out, but nature does the things the way because there's a reason. And if that simply happened, then we would not get NADH, right? Yes. So if OA just came out, we would not be able to generate the NADH. And because the cytosolic NADH is very low, simply OA coming out would not help gluconeogenesis, right? So, but you can go to malate, then malate come out. So it's basically reduced it to malate, then malate comes out, then you can regenerate OA and produce NADH, which is needed in one of the reversible steps in glycolysis, right? Because now you're going back. So then you can use gluconeogenesis. The other thing is because you're siphoning malate out, you are slowing down CSE. So this is how body adjusts to the metabolic need. The need right now is to use glucose somehow, right? Whatever resource is available. So let's don't use CSE, slow it down and use the resource to make glucose.